So in the previous part, I have discussed about nuclear family receptors types. It is classified into two types. First one, type 1 receptor, which is present in cytosol, and type 2 receptors that are present in nucleus. So here we are discussing about type 1 receptor signaling today. Examples I have told that type 1 receptor signaling are estrogen receptors, glucocorticoid receptors, progesterone receptors, mineralocorticoid receptors. Those are present in cytosol, and as they are present in cytosol, so they act as homodimer, and they are bound with heat shock protein 90. And what are heat shock proteins? So heat shock proteins are the group of proteins that regulates the activity of nuclear hormone receptors. Those are basically molecular chaperones. So those chaperones that helps in protein folding and protects them from damage during stress. So here in this mechanism, the signaling mechanism, we take as an example of glucocorticoid receptors. You can see here glucocorticoid signals are crossing the plasma membrane and it enters into the cytosol. So it can cross the plasma membrane, that means it is a nonpolar signal. It is cleared, and it is, as it is nonpolar signal, so it can also cross the nuclear membrane, so it can binds to the receptors with the in the nucleus also. Glucocorticoid signals, as it enters into the plasma, it crosses the plasma membrane and enters into the cytosol. So here inactive glucocorticoid receptors are present. Inactive glucocorticoid receptors are present and it is bind with heat shock protein 90. As I have told that heat shock proteins are group of proteins that regulates the activity of nuclear hormone receptors. So those are chaperone proteins. So these are bounded with glucocorticoid receptors and make it inactive. And when the signal enters into the cytosol, then the proteins are dissociated. Here you can see that the HSP90 proteins are dissociated, and the signaling molecules are bind to the ligand binding region. As I have told before, that ligand binding region is the region where ligand binds with the receptors. So ligand binding region, in the ligand binding region, the glucocorticoid signals are bound, and those receptors has already NLS, means nuclear localizing region. This also I have told before, that nuclear localizing sequence or signal is an amino acid sequence that tags proteins. It permits the proteins to enter into the nucleus, and thus after dissociation of HSP90, the receptor is activated, and the receptor is activated. After activation, it dimerizes. It is actually cloning, and then it enters into the nucleus, and during entering, it exposes its NLS, means nuclear localization signal, which permits it to enter into the nucleus. And thus after entering the nucleus, those molecules are activated by co-activator. Here co-activator is required. So this co-activator is HAT co-activator. What is the full form of HAT? Histone acetyl transferase. This HAT co-activator binds with the DNA. How it binds? These receptors has DNA binding region, and those DNA binding region have zinc finger motifs. So what is zinc finger motifs? That also I have told before in the previous part, that zinc finger motifs is a protein domain that controls gene expression by binding with DNA. So here we can see that it binds with DNA, and where it binds? In the hormone response element site. So hormone response element site is a site in the DNA sequence that binds to the hormone receptors to regulate the gene expression. So those zinc finger motifs bind to the DNA in the hormone response element site, and this we can see here. After the binding, it shows target genes expression, and the target gene and for the target gene expression successfully, the DNA must be euchromatin DNA. We all know that euchromatin and heterochromatin. What is the difference between euchromatin and heterochromatin? If the DNA is in heterochromatin, then it is more condensed condition, and if it is euchromatin, then it will be less condensed. So in less condensed condition, it will be easy to bind to the to bind with the DNA for the receptor.